The overall goal of this experiment is to determine NK cell count and cytotoxic activation levels in a human whole blood sample in a fast, reliable, and stable manner.
This method can help answer questions in the immunological field, such as what are the effects of outside interventions on the bioactivity of NK cells?
The main advantage of this technique is that it is fast and accurate, while still providing reproducible results across experiments.
To isolate NK cells from human whole blood, first place 15 milliliter tubes labeled whole blood sample, negative fraction, and positive fraction into the appropriate positions in the cell separator tube rack. When all of the tubes are loaded, insert the separator rack onto the mini sampler for auto-labeling, and select Reagent on the menu to highlight the position where the vial will be placed on the rack. Select Read Reagent to activate the 2D code reader, and hold the appropriate reagent vial in front of the 2D code reader at an angle 0.5 to 2.5 centimeters from the code reader cover. Place the read vial into the appropriate separator rack position. Next, under the Separation tab, highlight the desired positions. Then open the Labeling submenu and select an auto-labeling program. Assign the cell separation reagent microbeads to rack positions 1, 2, 3, and 4, and select the post-cell web separation program. Select the Rinse Wash program. Enter 1500 microliters for the sample volume under the volume.